Good day, everyone. I'm Someshwar, and today we're diving into the world of biosensors and their applications in healthcare and biotechnology. Biosensors are like our body's translators, converting biological responses into measurable signals. They're shaping a future where diseases are detected early and treatments are personalized. Throughout this lecture, we'll explore the principles, types, and transformative impact of biosensors in improving human health and advancing biotechnology. Let's start with the basics. Biosensors represent an intersection of biology and technology. So, they are basically analytical devices designed to detect and measure specific substances or entities within a sample. So, what sets them apart from other sensors is that their utilization of biological components to achieve this task. Every biosensor consists of a biological sensing element. So, these could be enzymes, nucleic acids, antibodies, or even whole cells. This is selected for their ability to interact with the target analyte. So, an analyte is the substance or entity that we're interested in detecting. This interaction is crucial because it triggers a biochemical reaction that ultimately generates a measurable signal. Now, this signal can further be amplified, it can be filtered, and it can be fed into a system where it displays on a screen. Let's think of it this way. When the biological element encounters the target analyte, it's like a key fitting into a lock. So, the interaction might cause a change in electrical conductivity, optical properties, or some other physical characteristics that can be measured. It's this transformation of biochemical signals into measurable outputs that forms the basis of biosensor technology. Let's look at the importance of biosensors in healthcare. Um, Biosensors can be used for early disease detection. Biosensors enable rapid and sensitive detection of specific molecules, aiding in early disease diagnosis and management. It is used in glucose monitors where the blood glucose levels are monitored continuously and it determines whether the patient is diabetic or not. Based on this, insulin can be provided to the patient. Um, Biosensors are used in monitoring biomarkers, so they help in continuous monitoring of biomarkers for various diseases, facilitating personalized medicine. They're mainly used in drug discovery. Biosensors play a crucial role in drug discovery by facilitating high throughput screening and analysis of drug target interactions. High throughput screening is a method used in the drug discovery to rapidly test thousands of compounds for their ability to interact with a particular target, such as a protein or enzyme involved in the disease process. Biosensors enhance this process by enabling the quick and efficient detection of these interactions. So, by measuring these signals, researchers and scientists can determine whether a particular compound shows a promise as a drug candidate or not. Biosensors enable this detection to be automated and conducted in a large scale, allowing researchers to screen thousands of compounds in a short amount of time. Importance of biosensors in biotechnology. It is used in environmental monitoring, where biosensors are used for real time monitoring of pollutants and contaminants in the environment. They provide very high sensitivity, enabling the detection of pollutants at very minute levels, at trace levels, crucial for assessing environmental health. Biosensors facilitate early warning systems for pollution events, allowing prompt intervention to mitigate environmental change. Biosensors are used in food safety, so they can enable rapid detection of pathogens and contaminants in food products, ensuring the food safety. It is also used in industrial processes, whereas it is used in optimizing industrial process by providing real-time monitoring of parameters such as pH, temperature, and substrate concentration. This enhances process efficiency and product quality. Um, Biosensors contribute to reducing production costs and minimizing waste by enabling timely adjustments to process conditions. Furthermore, They facilitate predictive maintenance by monitoring the equipment performance. This helps prevent costly downtime and equipment failures. Integration of biosensors into industrial automation enhances process control and enables data driven decision making, driving overall operational excellence and competitiveness. Let's look at the evolution of biosensors. The foundation of biosensors was laid with early designs relying on mainly enzyme based reactions. So, enzymes acted as a biological sensing elements, catalyzing reactions with target analytes to produce measurable signals. So, these early biosensors were instrumental in demonstrating the feasibility of using biological components for analytical purposes. So, this formed a base. Um, Expansion of biological elements. So, over time, biosensors evolved with the incorporation of additional biological elements such as antibodies, nucleic acids, even whole cells. The expanded this expanded their applicability, enabling detection and quantification of a wider range of analytes. Antibodies offered very high specificity, while nucleic acids enabled nucleic acid-based detection methods like PCR amplification, significantly enhancing detection capabilities. Integration of advanced technologies. Biosensors underwent a transformative phase with the integration of nanomaterials, microfluidics, and advanced signal transduction technologies. Microfluidic systems, it enabled a precise control of sampling handling and analysis. Advanced signal transduction technologies. This included electrochemical, optical, and piezoelectric methods. So this offered improved detection and selectivity. Biosensors exist in various forms to cater to diverse application needs. These days, wearable biosensors enable continuous monitoring of physiological parameters such as glucose levels or heart rate, enhancing personalized healthcare. These are the main components of a biosensor. A biosensor mainly consists of a bioreceptor, a transducer, and some sort of signal processing, so it can be read by microelectronics. The bioreceptor is able to detect the analyte, which are usually enzymes, antibodies, DNAs, and cells. So this sends either an electrical, optical, heat, or mass signal to a transducer. So the transducer receives the signal, and which is later amplified and filtered to be used with microelectronics. Let's look into these components a bit more. One, so biological recognition element. So this is the part of the biosensor that interacts with the target analyte, which is the enzyme, either the antibody, nucleic acid, cell receptor, etc. Transducer. The transducer converts the biological response triggered by the interaction between the recognition element and the analyte into a measurable signal. Common types of transducers include electrochemical, optical, piezoelectric, and thermal transducers. Number three, signal processor. So the signal processor amplifies, filters, and processes the signals generated by the transducer. So this can be used to enhance its detection and accuracy. It may also include components for data acquisition and analysis. Number four, we have an output display. This is the display that the user views. The output display presents the processed signal in a human readable format, such as a digital readout, graph, or a light, even an indicator light. Sample handling system. This component facilitates the introduction of the sample containing the analyte to the biosensor. It may include the sample preparation and delivery mechanisms, such as microfluidic channels, sample holders, or membranes even. We look at enclosure and interface. The biosensor may be enclosed in a casing to shield it from environmental factors, and this ensures its stability and longevity. Additionally, it may have interfaces for connections to external devices for data transmission and etc. Here we have a brief overview of the topics we just looked at. So here we have an input signal which is detected by the sensor and the transducing element. So the transducing element outputs a certain signal that can be amplified and filtered to be used with a system along with a display so it can be viewed by the user. Biosensors, the two components that we're going to look into deeper are bioreceptors and transducers. Bioreceptors can detect enzymes, antibodies, DNA, cells, etc. Similarly, transducers can output electrochemical, piezoelectric, calorimetric, and optical outputs. So, transducers are pivotal components in the realm of biotechnology and medical devices, serving as crucial intermediaries that translate the biological signals into measurable outputs. Their functionality is paramount for detecting, monitoring, and comprehending biological processes. Let's look into this a bit deeper. Functionality. Transducers serve as indispensable bridges between biological and measurable worlds. They receive inputs in various forms, such as electrical impulses from neurons, chemical signals from molecules, or even mechanical forces from tissues. And they transform these biological signals into quantifiable outputs that can be read by us. These outputs typically manifest as electrical voltage, current variations, or digital signals, which are then ready for analysis. There are different types of transducers. Number one, electrodes. These transducers are instrumental in measuring the electrical signals generated by biological entities. For example, EEG electrodes are used to monitor brain activity, and ECG electrodes are used for monitoring heart function. Number two, biosensors are designed to detect chemical compounds within biological samples. Biosensors are a vital category of transducers. Glucose sensors, for instance, they determine the glucose levels in blood samples. So this offers crucial insights for diabetic patients. Number three, we have mechanical transducers. These devices monitor mechanical changes within biological systems. For example, force sensors integrated in prosthetic limbs can detect pressure changes, and this enables users to control movements with precision and accuracy. So, by effectively converting biological signals into measurable outputs, transducers empower researchers, engineers to gain valuable insights into biological processes. Recognition elements are specific molecules that bind to target molecules with high affinity and selectivity. They are fundamental components of biosensors, diagnostic assays, and other biomedical technologies for detecting and quantifying analytes of interest. Let's look at some common recognition elements and their roles. Antibodies. Antibodies are formidable proteins manufactured by the immune system in response to foreign invaders. So these are known as antigens. With their remarkable specificity and affinity for target antigens, antibodies are indispensable in immunoassays for detecting biomolecules. So this includes proteins, hormones, pathogens, etc. They exist mainly in two forms, which is monoclonal, featuring identical copies, or polyclonal, which comprises a mixture of different antibodies. Secondly, we have enzymes. Enzymes are the biological catalysts in chemical reactions. So they are frequently harnessed as recognition elements in biosensors. By catalyzing reactions involving the target analyte, enzymes induce the generation of detectable signals. Uh, a principal, exploited in techniques like enzyme-linked immunoabsorbent assays, which is the short form is ELISA, and enzymatic biosensors. These enzyme-based detection methods offer exquisite sensitivity and specificity in detecting analytes of interest. Third, we have nucleic acids. Nucleic acids encompassing DNA, RNA emerge as versatile recognition elements in biosensors and diagnostic assays. Techniques such as PCR, which is polymerase chain reaction, and nucleic acid hybridization facilitate the detection and quantification of specific DNA and RNA sequence. So this enables applications ranging from genetic testing all the way to pathogen detection and forensic analysis as well. Next, we have aptamers. Aptamers are single-stranded DNA or RNA molecules which are crafted through the systematic evolution of ligands. So these are deployed as recognition elements in biosensors, drug delivery systems, therapeutic agents, etc. Now, these aptamers, they offer a very compelling alternative to antibodies, demonstrating remarkable versatility and adaptability across various biomedical applications. Next, we have peptides and MIPs, which are molecularly imprinted polymers. Peptides and MIPs represent synthetic recognition elements, which are meticulously engineered to mimic the binding properties of antibodies and enzymes. So recognition elements basically embody the essence of biosensing technologies, enabling the precise identification and quantification of target analytes, critical for biomedical research, clinical diagnostics, etc. The main types of biosensors include electromechanical biosensors, optical biosensors, piezoelectric biosensors, thermal biosensors, and nanotechnology enabled biosensors. Let's dive a bit deeper into these. Let's start off with electrochemical biosensors. Now, the main principle of working is these electrochemical biosensors, they utilize the conversion of biological recognition events into electrical signals. They typically consist of a biological recognition element such as enzyme, antibody, DNA. So this is immobilized on an electrode surface. There are three main types of electrochemical biosensors. Number one, we have amperometric biosensors. So these measure currents produced by a redox reaction at a constant potential. Second, we have potentiometric biosensors. These measure potential difference or voltage generated by the binding of analyte to the recognition material. Third, we have conductometric biosensors. So they measure changes in electrical conductivity due to the interaction between the analyte and the recognition elements. There are several applications of electrochemical biosensors. They are mainly used in fields including medical diagnostics, environmental monitoring, food analysis, bioprocess control, etc. Our next type of sensor is known as an optical biosensor. So the principle of working is that these optical biosensors, they detect changes in light proportions such as absorbance, fluorescence, Ri, which is refractive index, upon interaction with the analyte. There are various types in this itself. So absorption based biosensors. This measures changes in absorbance of light due to the analyte binding. We have fluorescence based biosensors, which utilizes the changes in fluorescence intensity or lifetime upon analyte binding. Next, we have surface plasmon resonance, which is SPR biosensors. They measure changes in the refractive index at a metal surface due to biomolecular interactions. There are several applications for optical biosensors, main one being medical diagnostics. Optical biosensors play a crucial role in medical diagnostics by detecting biomarkers associated with various diseases. They can detect proteins, nucleic acids, hormones, and other molecules present in bodily fluids like blood, saliva, or urine. These biosensors are used for early disease detection, monitoring disease progression, and assessing the effectiveness of treatments. For instance, they are again used in glucose monitoring for diabetic management, More recently, they've been used for COVID 19, monitoring COVID 19 as well. Next, we have piezoelectric biosensors. The main working principle is that they detect the changes in mass or viscoelastic properties of the sensing layer upon analyte binding. This leads to a change in the resonant frequency of piezoelectric crystals. When a voltage is applied to the crystal, it oscillates at its resonant frequency. Now, analyte binding, it causes a shift in this resonant frequency, which is proportional to the analyte concentration. So the concentration can be measured based on this shift in frequency. They are commonly used in medical diagnostics, again environmental monitoring, food safety, due to their high sensitivity and real monitoring capabilities. Next, we have thermal biosensors. So these biosensors, they measure changes in temperature resulting from biochemical reactions occurring on or near the sensor surface. Upon analyte binding, the heat released or absorbed by the reaction, it alters the temperature of the sensing element. So this change in temperature is detected by the transducer. The main applications of again thermal sensors are in medical diagnostics for detecting biomolecules such as proteins and nucleic acids. It is also used again in food safety and environmental monitoring. The next type of sensors are nanotechnology enabled sensors. These are the latest and greatest types of sensors. It plays a crucial role in detecting the sensitivity, selectivity, and functionality of biosensors. Nanomaterials such as nanoparticles, nanowires, nanotubes are all integrated into one package for the biosensors to improve performance. Now, the advantages of using nanotechnology enabled biosensors are that these materials, these nanomaterials, they provide a large surface area for biomolecule immobilization. They facilitate signal amplification, enhance biocompatibility. They also enable multiplex detection. Now, again, the common applications of these nanotechnology sensor biosensors are, they are included in medical diagnostics, POC testing, which is point of care testing, environmental monitoring, food safety. It is mainly used in food safety due to the high sensitivity and specificity. We realized that biosensors are mainly used in 6 areas, which is healthcare, environmental monitoring, energy harvesting. It's used in the food industry. It is used in electronics and agricultural purposes. We will look a bit deeper into the applications in healthcare and the applications in biotechnology. So, the applications of biosensors in healthcare are disease diagnosis and monitoring, point of care testing, which is POCT, wearable biosensors for continuous health monitoring, drug discovery and development. Biosensors play a crucial role in disease diagnosis and monitoring by providing rapid, sensitive, and specific detection of biomolecules associated with diseases. Here are some key roles that they play. Number one, early diagnosis. Biosensors they enabled early detection of diseases by detecting biomarkers present in bodily fluids or tissues. Early diagnosis is very critical for effective treatment and management of many diseases, including cancer, diabetes, and infectious diseases. Second, monitoring disease progression. So, biosensors can be used to enable continuous or frequent monitoring of disease progression by measuring changes in biomarker levels over time. This is particularly used for chronic diseases like diabetes, cardiovascular diseases, where regular monitoring is necessary for effective management. Third, it is used in personalized medicine. Biosensors they contribute to the development of personalized medicine by providing real-time information about an individual's health status. This information can be used to tailor treatment plans according to the specific needs of each patient. This improves the treatment outcomes and reduces the adverse effects of the disease. It can aid in remote monitoring. Now, with recent advancements in technology, um, biosensors can be integrated into wearable devices or implanted sensors. There are now implanted sensors for continuous remote monitoring of patients. This allows healthcare and professionals to monitor the patient's health status in real time, even from a distance, and intervene promptly when necessary. It is used in drug efficacy monitoring, where the biosensor is used to monitor how efficient the drugs are working in real time by measuring the changes in the biomarker levels before and after the treatment. So this enables healthcare providers to have a good baseline of what the drug or how effective the drug is on a certain person or on a certain treatment. So based on this, it is possible to adjust the treatment regimens and ensure the optimal therapeutic outcomes. It is used in detecting drug resistance, where it is by monitoring the changes in biomarker patterns over time. It can be used to detect resistance in infectious agents such as bacteria or viruses. Biosensors can again help identify emerging resistance and guide to the selection of appropriate antimicrobacterial therapies. Point of care testing, or POCT, revolutionizes healthcare delivery by bringing diagnostic testing closer to the patient. This bypasses the constraints of centralized labs by using the biosensor's unique capabilities. First, biosensors are known for their portability. Unlike bulky lab equipment, biosensors can be used portably, as they are very compact and lightweight, making them easily transportable for various healthcare settings, including bedside consultations, outpatient clinics, and remote locations. This mobility empowers healthcare providers to conduct tests swiftly and efficiently right where the patients are. Moreover, biosensors, they boast rapid response times, a critical attribute for POCT. Traditional lab tests may require hours or even days for results, actually. This causes delays in treatment's initiation. In contrast, biosensors, they deliver rapid results. This enables healthcare professionals to make informed decisions quickly and promptly. Um, Biosensors are designed for ease of use, making them accessible to broader range of healthcare providers, including nurses and primary care physicians. These devices, they typically feature intuitive interfaces and simplified procedures. This minimizes the need for any sort of extensive training. It can be used by everyone. Um, Even in resource limited settings where specialized lab expertise may be scarce, it can be used for confidently acquiring information. One compelling example of biosensors in POC is the widespread application in diagnosis and management of diabetes. Glucose biosensors, for instance, they play a pivotal role in monitoring blood sugar levels. These sensors, they enable diabetic patients to perform regular self-monitoring of blood glucose levels. So they don't need the company of medical professional. It can be done by themselves. So moreover, CGM, or continuous glucose monitoring, it provides real-time glucose readings, offers insights into glucose functions, fluctuations throughout the day. This continuous monitoring, it helps individuals with diabetes and their healthcare providers to optimize treatment strategies and improve long-term outcomes. In essence, biosensors in POCT are a new era of personalized and proactive healthcare. Next, we have wearable biosensors for continuous health monitoring. Wearable biosensors are integrated into wearable devices such as smartwatches, fitness trackers, and patches. This allows for continuous monitoring of various health parameters in real time. Now, these biosensors they measure physiological signals, biometric data, and biochemical markers directly from the wearer's body itself. This provides valuable insights into their health status and activities. Continuous health monitoring with wearable biosensors enables early detection of health issues. It provides personalized health management and remote patient monitoring as well. Excuse me. One example of wearable biosensors, it can monitor vital signs such as the heart rate, BP, body temperature, oxygen saturation levels. They can also track physical activity, the number of steps you've taken, your sleep patterns, your stress levels, etc. Continuous monitoring of these parameters, they help individuals and healthcare professionals assess overall health and detect any sort of abnormalities or changes that may indicate any sort of underlying health conditions. Another common application of biosensors in healthcare would be drug discovery and development. Now, biosensors they play a very significant role in drug discovery and development by enabling high throughput screening of potential drug candidates. Traditional methods of drug screening can be very time consuming and costly. Biosensors they streamline this process by allowing researchers to rapidly screen very large libraries of compounds for potential drug candidates. Now, this high throughput capability it accelerates the pace of drug discovery. This enables the researchers to identify promising compounds more efficiently. Now, studying drug target interactions. Understanding how drugs interact with their molecular targets is very crucial for optimizing their efficacy and minimizing side effects. Biosensors they provide a means to study these interactions in real time and with high sensitivity. By monitoring parameters such as binding affinity and kinetics, researchers can gain insights into the mechanism of action of potential drugs. They are used also in monitoring cellular responses, where it allows researchers to assess their effects on various cellular pathways and processes. This information again is very valuable for evaluating the therapeutic potential of drug candidates and predicting their clinical outcomes. Next, under drug discovery would be using uh, pharma, uh, uh, pharmacokinetics and pharmacodynamics, which is short form is PK and PD. Biosensors again they play a very vital role in PK and PD. They can measure drug concentrations in biological samples with high precision and sensitivity. This provides valuable data on drug absorption, distribution, metabolism, and even excretion. This information is essential for optimizing uh, drug dosing regimens, and ensuring therapeutic efficacy. Biosensors can be used for toxicity screening. So, by monitoring biomarkers of toxicity in real time, biosensors can help identify potential adverse effects, so potential negative effects early on in the drug development process itself. So, this allows again researchers to modify or eliminate toxic compounds before they progress into clinical trials, before they progress into the hands of the users. They are used for optimizing these drug formulations. So over time, biosensors are used to uh, optimized by monitoring drug stability, solubility, and release kinetics. So based on these information that we collect, we can have better formulations. We can come up with better drug formulations that are both effective and safe for patients. Next, again, personalized medicine and companion diagnostics. Biosensors, they contribute to the paradigm of personalized medicine by enabling the development of companion diagnostics. Next, we look at the applications of biosensors in biotechnology. They are used in environmental monitoring, food safety and quality control, bioprocess monitoring and control, detection of biomolecules in research. Environmental monitoring. Now, biosensors, they play a crucial role in environmental monitoring by detecting pollutants, pathogens, and other harmful substances in the air, water, and soil. They offer real-time monitoring capabilities, enabling early detection of contaminants, um, and facilitating timely interventions to prevent environmental degradations. Some examples of these include detection of heavy metals. Now, biosensors, they can detect heavy metals like lead, mercury, and cadmium in water sources. Now, these heavy metals in water sources are extremely polluting, and it enables monitoring of the pollution levels and ensuring compliance with the environmental regulations. Next, it is used for monitoring of pesticides and herbicides. Biosensors can again detect pesticides, herbicides, residues in soil and water. Now, this helps to assess the impact of the agricultural practices on the environment and ensuring food safety. Next, it is used for bacterial contamination detection. Biosensors equipped with specific antibodies or DNA probes can detect any sort of bacterial contaminants in environmental samples, in water quality and public health. In biotechnology, biosensors are used for food safety and quality control. Now, biosensors are widely used in the food industry to ensure the safety and quality of food products. They enable rapid and on site detection of contaminants, pathogens, and adulterants, thus minimizing the risk of foodborne illnesses and ensuring compliance with food safety regulations. Some applications include detection of foodborne pathogens. So biosensors, they can detect the pathogens such as Salmonella, E. coli, and Listeria in food samples. This provides a timely information to prevent contaminated products from reaching the consumers. It is used in identification of allergens. Now, biosensors can detect allergenic substances like gluten, peanuts, and soy in food products. This helps in preventing allergic reactions in sensitive individuals. Next, under food and safety, would be monitoring of food adulteration. Biosensors, they can detect adulterants of and contaminants in food products such as melamine in milk or fraudulent substitution of high value ingredients. This ensures the authenticity and the quality of the food items being provided. Number three, we have bioprocess monitoring and control. So biosensors, they are instrumental in bioprocess monitoring and control in various industries. So this includes pharmaceuticals, biotechnology, and fermentation. They provide real-time data on key process parameters, enabling optimization of production processes and ensuring consistent product quality. Some examples of bioprocess monitoring and control are as follows. Uh, Monitoring of biomolecule concentrations. Now, biosensors, they can monitor the concentration of biomolecules such as glucose, lactate, amino acids, bioreactor systems, facilitating process control, optimization in biopharmaceutical production. And another example of bioprocess monitoring control would be in the detection of metabolic activity. So biosensors, they're equipped with microbial or enzymatic sensors They can detect metabolic activity and substrate consumption in fermentation processes. Now, this allows for a very precise control of fermentation parameters and maximizing product yields. Um, Another example would be online monitoring of cell viability. Now, biosensors can monitor cell viability and metabolic activity in cell culture systems. This provides valuable information for cell culture optimization and bioprocess scale up. The last topic under applications of biosensors in biotechnology would be the detection of biomolecules in research. Biosensors, they are indispensable tools in biomolecular research for the detection and quantification of various biomolecules, again including proteins, nucleic acids, and small molecules. They offer a very high sensitivity, specificity, and throughput, enabling a wide range of applications in biomedical research. Some examples of detection of biomolecules in research would be protein detection. Biosensors, they can detect proteins with very high specificity and sensitivity. This makes them valuable tools for protein quantification, biomarker discovery, and protein interaction studies. They are used in nucleic acid analysis. Biosensors can detect nucleic acids, DNA, and RNA through hybridization or amplification based approaches. This facilitates applications such as gene expression analysis, genotyping, and pathogen detection. Some biosensors can also detect small molecules such as hormones, neurotransmitters, and metabolites. So, this enables studies on cellular signaling pathways, drug discovery, and disease diagnostics. This is a small recap of the topics we have covered under applications of biosensors. So, namely, the detection of infectious diseases, soil management, prosthetic devices, food quality assurance and management, drug discovery, environments mapping and monitoring, toxin detection in environment lab factory, water quality identification management, and mapping. There are some challenges and future perspectives of biosensors which we shall discuss. Biosensors, they have revolutionized various fields such as healthcare, environmental monitoring, food safety, biodefense due to their ability to detect and quantify biological and chemical substances with very high specificity and sensitivity. However, despite these significant advances, biosensors, they still face several challenges which we shall discuss. So, sensitivity and specificity improvement. Achieving higher sensitivity and specificity is crucial for enhancing the performance of biosensors. This involves improving the signal to noise ratio and minimizing any cross reactivity with non target molecules. Strategies such as utilizing novel recognition elements such as aptamers, molecularly imprinted polymers, optimizing surface chemistry, and incorporating signal amplification techniques such as enzymatic amplification, nanoparticle labels. This can enhance sensitivity and specificity. Now, advancements in nanotechnology and material science have enabled the development of nanostructured surfaces and nanomaterial based signal transduction platforms. This further improves the sensor performance. Another challenge would be miniaturization and integration. Miniaturization of biosensors, it allows for a portability, ease of use, and integration into various devices including wearable devices, point POC diagnostics, and lab on chip platforms. Microfabrication techniques such as photolithography, soft lithography, and 3D printing enables this fabrication of miniaturized sensor components with very high precision and throughput. Cost reduction and scalability. This is one of the significant barriers to using biosensors is the high cost. So the high cost is a significant barrier to the widespread adaptation of biosensors, particularly in resource limited settings. Therefore, efforts are focused on reducing the overall cost of biosensing devices while maintaining very high performance. So, utilization of low cost materials and fabrication techniques such as paper based sensors and printed electronics can significantly reduce production costs. Scalability of manufacturing processes is very essential to meet the increasing demand for biosensors. Techniques like roll-to-roll printing and mass production methods enables large-scale fabrication of sensors at low cost per unit. Some emerging trends in biosensor technology are the multiplex detection. So the ability to detect multiple analytes simultaneously is increasingly important for comprehensive analysis in various applications. Multiplexed biosensors can enable high throughput screening and diagnostics. Again, POCT, point of care testing. This is a growing demand for biosensors that can be used at point of care, providing rapid and on-site detection without the need for centralized lab facilities. POC biosensors enable real-time monitoring and timely intervention in healthcare settings. Recently, IoT or integration in Internet of Things integration is with IoT platforms. This enables remote monitoring, data transmission, real-time analytics, um, personalized healthcare, environmental monitoring, smart agriculture, etc. AI, artificial intelligence, and machine learning algorithms are being employed to analyze complex sensor data, improve detection accuracy, and enable predictive analytics. So, AI driven biosensors often enhanced performance and adaptability in dynamic environments. So let's end this lecture with a small example of the working principle of a glucometer. As throughout the lecture we have mentioned glucometer multiple times. Let's just dig a bit deeper into the working of glucometer. Now, a glucometer with a biosensor is a device. It's used to measure blood glucose levels, primarily for people with diabetes. So the process starts off with a sample collection. So this process of obtaining blood, small sample of blood, usually by pricking the fingertip with a lancet or create a small drop of blood. So this blood is then applied to a test strip. This strip contains the biosensor itself. The test strip, it consists of several layers. Now, the first layer is the application layer, second is the reaction layer, and third we have an electrode layer. So the reaction layer, which is the middle layer, it contains enzymes such as glucose oxidase, which reacts with glucose. Now the enzymatic reaction is as follows. The sensor it uses an electroenzymatic approach. This means that it takes advantage of the glucose oxidation with the glucose oxidase enzyme, which is the enzyme we have used. So the presence of glucose oxidase, it catalyzes the chemical reaction of glucose with oxygen, which causes an increase in pH. So this decreases the partial pressure of oxygen and increase of hydrogen peroxide because of the oxidation of glucose to gluconic acid. So the chemical equation is glucose on reacting with oxidation on oxygen along with glucose oxidase enzyme produces gluconic acid and H2O2, which is hydrogen peroxide. So the test strip, it measures changes in one or several of these components to determine the concentration of glucose. The strips used in this design have mainly three terminals or electrodes, as shown here. So there is a trigger electrode, there is a working electrode, and there is a reference electrode. Now, a negative voltage of minus zero point 0.4 volt is applied at the reference electrode. So, when a blood or glucose solution is placed in the strip, a chemical reaction occurs inside it. This generates a small electrical current which is proportional to the glucose concentration. This current is constantly monitored using a device in which the strip is placed in. Once the strip is placed in, we wait for the chemical reaction to stabilize for around 5 seconds, and the voltage which is read by the ADC, it is compared to a table which is readily available. So this table contains values of glucose and the voltage read by the ADC. So we can obtain the proportional glucose value based on the voltage obtained. So this value is then sent to a computer and later to a screen or a display for the user to view it. Now, the electrical current produced by the enzymatic reaction is measured by the electrodes, which is embedded in the test strip itself. So these electrodes, they are typically made of conductive materials such as gold or carbon. Now, the main process would be signal processing. The signal generated by the enzymatic reaction is very weak. So it needs to be amplified in some way and processed to obtain an accurate glucose measurement. The glucometer, it contains a microprocessor and electric circuitry for signal amplification, noise reduction, and calibration. So before amplification, we need to first convert the current to voltage. So the output generated by the test strip is a current that represents the glucose concentration. This current must be converted to voltage so that it can be properly filtered and treated. This conversion is performed using a current to voltage converter that has a single supply, a low input offset voltage, low input offset, and bias current triamp. The formula for the output voltage would be V out is equal to the input current from the sensor itself into the resistance. Now this is an example of an amplified and filtered circuit. The block is divided into an amplification section and then a low pass filter with a cutoff frequency of 8 Hz designed to eliminate high frequency noise. The equation for the filter cutoff frequency would be 1 by 2 pi RC, where R is the resistor and C is the capacitor. Finally, we would have a display which outputs the processed glucose measurement. So this display usually shows the glucose concentration in milligrams per deciliter or millimoles per liter. So it depends on the user's preference. We have now reached the end of the lecture, and I would like to thank my fellow students and my professor for this opportunity. Thank you.